My name is Lindsay, and I'm a postdoctoral researcher at the Centre for Clinical Microbiology at UCL. I'm currently working part-time as a site coordinator for the clinical trials that we're part of, looking at improving drug treatment for tuberculosis. I've recently done site visits at the Centre de Recherche Médicale de Lambaréné in Gabon and Kazumu University of Health Sciences in Malawi, and we want to show you what a typical day looks like for the laboratory teams who process all of the clinical trial samples. At any one time, there may be many different patient samples to process, and there are lots of different stages of the processing cycle. First, a patient who is part of the clinical trial comes to the clinic and gives a sputum sample. These are then transported to the lab in a cool box. Sputum samples are given their laboratory numbers, and all of the paperwork associated with those samples are filled in. Next, the samples are taken into the high containment laboratory. Tuberculosis is an airborne pathogen, so we must work in a cabinet to protect the worker from inhaling tuberculosis bacteria. The sputum is decontaminated with chemicals to kill any bacteria other than tuberculosis because we want a pure sample. The sputum is spun down in the centrifuge, and the pellet of bacteria at the bottom of the tube is used for further tests. Part of the sputum pellet is placed on a microscope slide and stained so that the bacteria show up. The team can then count how many bacilli they see, which indicates how heavily somebody is infected. Some of the sputum pellet is inoculated into a midget tube. This contains liquid media that the bacteria like to grow in. It can take many days or even weeks for tuberculosis to grow, as it's a very slow organism. Once the midget tube has flagged positive for bacterial growth, the team then do other tests to confirm that it's tuberculosis and not contamination from other bacteria or fungi. They plate out some of the culture onto an agar plate, and TB can't grow on blood agar, so this should remain clear. If it's contaminated with something else like E. coli, the other bacteria will grow, and usually within 24 hours. The Gene Expert or MPT64 molecular test, which uses the bacteria's DNA, are also used to confirm that it's Mycobacterium tuberculosis and not another similar Mycobacterium species. Once all these tests are done, we can confirm whether the patient still has active TB or not, which means we can identify whether the drugs have worked and killed the bacteria or not. Patients come around once a week to give a sample for the duration of the study. This means a lot of samples for the teams to process. For more information about our clinical trials and what we do, visit the Centre for Clinical Microbiology website.